In this lecture, we are going to discuss forensics, specifically how DNA is analyzed in forensics. At the end of the lecture, you should be able to list the uses for DNA forensic identification, describe the appropriate ways to use DNA in a criminal investigation, describe the theory of DNA typing in forensics, specifically CODIS, analyze examples of DNA profiles of suspects to determine which suspect is most likely the individual involved in a crime based on the DNA evidence left at the crime scene. DNA is different from every other individual except for identical twins, and we've already discussed that, and you know that a person's DNA is the same in every single cell. So DNA from a white blood cell in the blood is the same as DNA in the skin as well as in the semen. DNA can identify if a suspect was at a crime scene, but it cannot tell when the suspect was there or for how long a suspect may have been at a crime scene. DNA evidence can be found even from samples that are decades old. However, not all DNA evidence will result in a usable DNA profile. Environmental factors can negatively affect the quality of DNA. For example, high temperatures, heat, sunlight, moisture, microorganisms all can degrade DNA and make it not usable. DNA analysis in forensics can be used to link a suspect to the evidence or eliminate a suspect, and it's similar to the use of fingerprints in a molecular epidemiological study. You can use DNA to identify a victim by using a comparison of DNA from relatives. You can also link evidence found at one crime scene to DNA from evidence at another crime scene, and also link crime scenes to the same perpetrator either locally, statewide, or nationwide. There are various examples of DNA that can be used at a crime scene. For example, using saliva left on a stamp from a stalker that's sending out threatening letters, or skin cells that are shed on a ligature of a strangled victim, or perspiration on a baseball cap that's discarded at a crime scene, or a bite mark on a rape victim. Here's a chart of various types of DNA evidence, the location where you would most likely find your DNA, and the source of the DNA from that specimen. So, in a baseball bat, the source of the DNA is going to be sweat, so you're going to have skin cells, possibly white blood cells from a blood specimen, tissue cells, and you're usually going to find that DNA at the handle of the bat where someone was holding it, and possibly blood and tissue from the victim at the end of the bat where maybe that bat was used to beat someone. How does DNA forensic identification work? Any type of organism can be identified by examining DNA sequences that are unique to that species. To identify individuals, what's done is 13 DNA regions that vary, so variable regions, variable from person to person, are used, and these variable regions create a DNA profile of that individual, and this is also considered a DNA fingerprint. There's an extremely small chance that another person has the exact same DNA profile for these 13 specific regions. DNA was first allowed as evidence in 1990. DNA is going to be used to identify a potential suspect whose DNA may match evidence left at a crime scene. Also, DNA is going to be used to exonerate persons that are wrongly accused of crimes. There's been several people have been imprisoned for many years that have been let out of prison because of advances in molecular technologies. You can use DNA to identify a crime or a catastrophe victim, to identify endangered and protected species as an aid to wildlife officials. This could be used to prosecute poachers. You can use DNA to detect bacteria and other types of microorganisms that may pollute the air, water, soil, and food, and also determine pedigrees for seed or livestock breeding. So DNA identification can be very effective if it is used correctly. So the portions of the DNA sequence that vary the most among humans must be used. Also, these portions must be large enough to overcome the fact that human mating is not absolutely or may not absolutely be random. So, how do you use DNA in a criminal investigation? Accumulating bits of linking evidence in a chain of evidence. Each bit of evidence by itself is not very strong, but when you pull all of these accumulating bits of evidence together, you can actually then argue that your suspect is really the correct person. You're going to look for matches based on sequence or on numbers of small repeating units of DNA sequence, your short tandem repeating DNA sequences. You're going to look for these at several different locations on the person's genome. So, one or two matches are not enough to be confident that the suspect is the right one, but anything 4 or 5 might be used. A match at all 5 is rare enough to prove beyond a reasonable doubt that the right person is accused. So, DNA typing in a criminal case involves obtaining the sample, so that includes crime scene evidence and as well as finding potential suspects. So when you get crime scene evidence that may have DNA in it, you have to make sure you don't contaminate that DNA. So you have to collect the specimen very carefully. You would have to extract the DNA from the specimen and then analyze the DNA for the set of specific DNA regions or the markers. So only 1/10 of a single percent of DNA, about 3 million bases, differs from one person to the next. So what's used are the variable regions in human DNA to generate a DNA profile, and usually these DNA samples are obtained from blood, bone, hair, body tissues, anywhere where you can get a very nice DNA sample. In a criminal case, usually you're going to obtain samples from your crime scene evidence, extract the DNA, and analyze the markers, and we already said that. So scientists find markers in a DNA sample by designing small pieces of DNA or probes that are going to bind to complementary DNA in a sample, so your typical blotting technique. So, a series of probes bound to a DNA sample creates your DNA fingerprint. And this DNA fingerprint or profile is compared to other DNA profiles that are known to find a match. So, we already said one marker by itself is not unique to a specific individual. However, if 4 or 5 different markers are used, you increase the chance that another individual is not going to have the same profile. So, different technologies that are used in forensic investigations. Well, usually we're looking at short tandem repeat analysis, and the FBI uses a standard set of 13 specific short tandem repeat regions. And the odds that two individuals will have the same 13 loci DNA profile is approximately 1 in 1 billion. Also, you can do variable number tandem repeat analysis, where you have your DNA region where you have a short sequence that's repeated in tandem 100 or more times. Usually, these can be between 8 to 35 nucleotides sequence repeats. The number of repeats is going to vary from individual to individual. Also, some gender specific analyses can be performed. You can do mitochondrial analysis, so you extract your DNA from your mitochondria. Mitochondria are transmitted by the egg and not the sperm, so this can be used to study individuals related through a maternal or female line. Also, you can use mitochondria to examine DNA from samples that can't be analyzed through restriction fragment length polymorphism or short tandem repeat analyses. So, for example, biological samples that lack nucleated cellular material, such as hair, bones, teeth, things like that. Maybe samples that are degraded. So, millions of mitochondria are there per cell, and you can get mitochondrial DNA from much smaller amounts of a sample. So, if you don't have a large enough sample size to collect genomic DNA, you can extract mitochondrial DNA and do analyses of mitochondrial DNA. You can also do Y chromosome analysis, which is useful for tracing relationships among males. So you're going to analyze biological evidence involving multiple male contributors. So we've already discussed short tandem repeats and variable number tandem repeats, and there are specific regions in the DNA within the human chromosome that you have tandem repeats. So you have specific DNA sequences that repeat over and over and over, and the number of repeats is going to vary from one person to another person. So every human being has some VNTRs, and to determine if a person has a specific variable number tandem repeat, we're going to do a southern blot. So your southern blot is going to be probed through a hybridization reaction with your radioactive version of that variable number tandem repeat. The pattern that's going to result is what we refer to as a DNA fingerprint, and a given person's variable number tandem repeats come from genetic information that is obtained by the individual's parents. So he or she could have variable number tandem repeats inherited from his or her mother or father, or a combination. So because variable number tandem repeat patterns are inherited genetically, a given person's variable number tandem repeat pattern is more or less unique. So the more variable number tandem repeat probes that are used to analyze a person's pattern, the more distinctive and individualized that pattern or DNA fingerprint is going to be. So in order to detect variable number tandem repeats, you're going to isolate DNA, you're going to digest the DNA with restriction endonucleases, and you're going to do an RFLP, or restriction fragment length polymorphism, and look at a DNA fingerprint, and the sizes of the bands are going to vary depending on the number of repeats a person has. So remember, these repeats are inherited in a Mendelian fashion, and you're going to inherit from each parent. So for example, in variable number tandem repeat analysis, you can determine a biological offspring. So you have VNTR pattern from a mother and a father. You have daughter 1, you can see, has bands that match both mom and dad. However, daughter 2 is actually dad's stepdaughter and mom's biological daughter. So you can see for daughter 2, there are bands that match mom, but not bands that match dad because dad's not actually the biological father. And son number 1, this is the biological son. And again, you're going to have variable number tandem repeats that match both mom and dad. And the second son is actually an adopted son. So this son is not biologically related, so it's going to have completely different bands. It's not going to have bands that necessarily match either mom or dad. You can use PCR to look at variable number tandem repeats and look at band sizes. So the more repeats someone has, the higher the bands or that they're going to have. So you might have 31 repeats, which is going to give you a 642 base pair band, whereas if someone only has 18 repeats, they're only going to have a 434 base pair band at that specific locus. So you can use your southern blotting and use your probes to determine a DNA fingerprint for 2 individuals. And in this case, it's Fred and Norma. Fred and Norma are not blood relatives. You could tell they do not have any matching bands. So you could see Fred and Norma's bands, and then you could look at Bob and Ann, and either Bob or Ann is the child of Fred or Norma, and it's actually Bob that is the child of Fred and Norma because Bob has one band that matches Fred and one band that matches Norma. So we already said you inherit one band from each parent. Siblings can inherit the same bands or different bands. So you can look at David. David inherited the top band from mom and the other band from dad. And Katie inherited the bottom band from mom and the other from dad. You always need control lanes. So lane number 3 is a national control, and it must be run and it must be the expected size. If it's not the expected size, you cannot use the results. Lanes 4, 5, and 6 are unrelated individuals, so you shouldn't really get matching bands. One band matches a band with Bob, but every now and again you're going to have a matching band, but you shouldn't have all matching bands with your unrelated individuals. You can do paternity testing where you have 4 different children, a mom and a dad, and you want to see which bands of each child match. Each child should get 1 band from mom and 1 band from dad. So you're using a single locus probe. You want to figure out which child is least likely to be the biological offspring, and in this case, it's child 2. Why is it child 2? Because child 2 lacks a band from the father. So here you go, look at child 2. You could see child 2 has a band that matches mom, but not a band that matches dad. Child 1 has a band that matches mom and a band that matches dad. Same thing with child 3 and child 4. You can look at repeat analysis to determine a rape victim. So you're going to look at blood samples from the victim, blood sample from the defendant, a vaginal swab since this is a rape of the victim, and you need to separate a rape sample, your female fraction and your male fraction. So in this case, you see that the blood sample of the defendant does not match the male fraction of the rape victim. So this person is not guilty of committing this rape. So CODIS is the Combined DNA Index System. CODIS is a software program that operates at the local, state, and national level. and it's a database of DNA profiles from convicted offenders, unsolved crime scene evidence, and missing persons. It was developed by the FBI to identify perpetrators of violent crimes. There are 13 short tandem repeat loci that are used in CODIS, and these are all tetrameric repeat sequences. So you have your different tiers of CODIS. You have the local level or the local DNA index system, and this is operated by police departments or sheriff's offices. DNA profiles are originated at the local level and can be transmitted to the state and national levels. You have your state DNA index system, which is going to allow local laboratories within that state to compare DNA profiles. And you have your national DNA index system, and this is the highest level of CODIS and enables qualified state labs that are actively participating in CODIS to compare state to state DNA profiles. These are the 13 CODIS short tandem repeat loci. You can see the locus, the different loci, so D3S1358, the VWA, FGA, D8S1179, D21S11, D18S51, D5S818, D13S317, D7S820, D16S539, the TH01, TPOX, CSF, FIPO, and the AMEL. Again, your 13 CODIS STR loci with the chromosomal position. So the TPOX loci is located on chromosome 2, the FGA is located on chromosome 4. So these 13 loci are used for criminal analyses. So again, your single locus probes, these are going to occur at only one site in the genome, and these are the loci that are highly variable from person to person. So for example, the locus D7S280 is one of 13 CODIS short tandem repeat loci that's found on chromosome 7, and you could have 6 to 15 tetrameric repeats. So you can see GATA, over and over and over again. So what you're figuring out is the frequency of an allele pattern. So the frequency is going to vary between race and ethnicity. It's determined in many different populations, and you can estimate the frequency of a multi-locus DNA profile, and this is based on a reference population. You're determining probabilities, so the chance of having any particular two loci, the same two profile at two different loci. So you're going to multiply the percentages. So at allele A is 10% chance, at allele B it's a 5% chance. You're going to multiply those percentages, 0.1 times 0.05. So there's a 1% chance that two people are going to have the same genotype at that loci. So you're going to look at several different variable number tandem repeat loci, and you're going to calculate the frequency of the different VNTRs. So for example, a DNA profile, you have 3 different loci here. You have the genotype at each loci. So at D3S1358, you have 1518. At VWA, you have 1616, and FGA, a 1924. What's the frequency at each locus? 8.2%, 4.4, and 1.7. So at the D3S1358, someone that has a 1518 genotype at that locus, 8.2% of the population has that same genotype. At the VWA, a 1616, 4.4% of a specific population has that genotype. So the biological evidence is compared to victims or suspects, and you need to combine the frequency at all 13 loci. In this example, 3 different short tandem repeat loci are being analyzed, and this is done on a machine, and all of the loci are analyzed simultaneously. So in this specific example, the D3S1358, BWA, and FGA loci are being analyzed. So the length of the amplified DNA is shown from 100 base pairs to 280 base pairs at the top of the figure. The middle panels with multiple peaks are reference standards with known alleles for each short tandem repeat locus. The alleles for the 3 loci don't overlap. The lower panel shows the alleles for the victim's mother at the 3 loci. These alleles are compared by computer to reference standards, and they're labeled. Commercial kits, such as the Profiler Plus, are available for short tandem repeat analysis. These kits have fluorescent molecules covalently linked to the primer. So in this Profiler Plus PCR amplification kit that's available from Applied Biosystems, 9 short tandem repeats are analyzed using 3 sets of primers, and each set of primer has a different colored fluorescent label. And this is an example is shown here using 3 sets of your short tandem repeat primers, one labeled blue, one labeled green, and one labeled yellow. The red peaks are the standards, the DNA size standards, and this system is one of the many computer software systems that can be used for STR analysis. So here's an example of the frequency of a 5 locus DNA profile, and this is based on semen that was obtained from a rape victim. So what is done here is you match 5 different variable number tandem repeat loci, and the frequencies of each of the 5 loci are 1%, 2%, 6%, 10%, and 3%. So if you take these percentages and multiply all of them, 0.01 times 0.02 times 0.06 times 0.1, you get a frequency of 3.6 times 10 to the minus 8, which means that by chance there is 1 out of 27.8 million individuals that have that exact genotype at these 5 different loci. So collection and storage for DNA analysis of specimens obtained from a crime scene. So of course, sometimes you only have very tiny samples, so it might be one little speck of blood spatter from a wall. So you have to very carefully collect and store any type of evidence that you're going to use for DNA analysis. And there's an entire chain of custody that has to be followed regarding collection and how it's collected and in what containers it's collected and where it's brought, and everything has to be signed, and you have to keep a record of every piece of evidence. So since environments can negatively affect DNA quality, the DNA has to be appropriately stored. So any evidence where you might obtain DNA at any point in time, it might not be soon after collection, it may be a year or 2 after collection, you have to store that evidence properly. You also have to be very careful so that you don't contaminate that evidence with other DNA. So DNA from another source can get mixed with your evidence. So when you're collecting your sample, you wouldn't want to sneeze or cough while you're collecting the evidence, because then you're going to add your DNA to the evidence. So in order to avoid contamination, specimens are always collected wearing gloves. Analysis is done using disposable instruments such as disposable pipette tips and disposable plasticware. The area where the evidence is cannot be touched, since you don't want to contaminate any of the DNA evidence with your own DNA. We already said you don't want to talk, sneeze, or cough over the evidence while you're collecting the evidence or while you're testing the evidence. And you wouldn't want to touch your skin while you're collecting evidence or packaging up the evidence. All evidence has to be stored in paper bags, and this is done because plastics retain moisture and moisture can degrade DNA. So all evidence is stored in brown paper bags so that light can't get through, and all of the paper bag evidence is stored in a separate room that's nice and dry and is at a constant room temperature so that there's no fluctuations in temperature and no possibility of moisture getting in to damage the DNA. So we already know the advantages and disadvantages of molecular techniques, and they're the same for forensic analysis as they are for any other analyses. So molecular techniques provide enhanced analytical specificity and sensitivity. You're also able to directly identify your DNA or RNA from your specimen. You could detect single cells. You could detect as few as 50 copies of nucleic acid, and your genotyping will allow you to detect variations in base sequences, which is the mainstay of forensic testing, where you are looking at the variations in tandem repeats in certain loci of the DNA. The limitations of molecular testing is that the cost is high. There is that risk of contamination while you're collecting evidence and while you're testing any evidence. Also, you are going to need additional lab space, especially in a crime lab, because there's the chain of custody, and you have to make absolutely sure that there's no contamination. So evidence is stored in one place, evidence is taken out of the bags and processed in another room. DNA extraction is done in another room. All amplification procedures are done in a separate room. You need special equipment for your molecular tests, all these special software that's needed. You need your safety apparel, your gloves, your disposable gowns, things like that. And you also need properly trained personnel. I'm going to go over a couple of cases that have used DNA for solving it. So here is just kind of a fun case, and this will make more sense when you go to the website. So on the evening of November 1st at approximately 8:15 p.m., Jimmy Sweet entered his bedroom, walked to his desk to sit at his computer. When he went to turn his computer on, he noticed that one item on his shelf was out of place. He looked a little closer, and sure enough, the object was disturbed. The object had previously been sealed in an airtight package, which was now ripped open. The object was still inside, although it had been altered. So what happened was this was Jimmy Sweet's lollipop, and someone opened up the lollipop and put it in their mouth and ate half of it and put it back in the package. So, of course, the prime suspects are Jimmy's 7 sisters: Candy, Cookie, Sugar, Lolly, Honey, Brandy, and Carmela. Each one of his sisters loves lollipops, so each one of them would possibly be of the person that ate half of it. Jimmy detained his 7 sisters, and DNA fingerprints are available for each one. So in this case, there's saliva that's left on the lollipop. So you can take that lollipop and extract the DNA from the saliva that was left on the lollipop. It's not actually the saliva that has the DNA, it's the mucosal epithelial cells from the mouth that is going to mix in with the saliva and get on the lollipop. So you're going to have to analyze the DNA left on the lollipop to determine which one of the sisters committed the crime. So here's the website, and I highly recommend you go to this website. It has a lot of information on, forensics and fingerprinting, and there's actually a computer lab where you actually perform a fingerprint and you figure out which one of the sisters committed the crime. So you should go to this website and really look at all the interesting things on this website. Okay, now this is an actual real published case, a previously unsolved case. So in August of 1994, a man broke into an apartment and raped a 24-year-old female that was sleeping in her apartment in Sacramento, California. Now, in the summer of 2000, the Sacramento detective that worked on the case happened to see an article about a warrant against a suspect named John Doe in 3 rape cases that occurred in Milwaukee. The suspect was identified by the DNA profile that he left at the crime scene. So in this case, an unknown male with a short tandem repeat DNA profile at the following genetic locations. So at each location, you can see the genotype. So at the D3S1358 locus, the genotype is 1515. The TH01 locus, the genotype is 77. So this person's DNA profile at these loci would occur in approximately 1 in 21 sextillion of the Caucasian population, 1 in 650 quadrillion of the African American population, and 1 in 420 sextillion of the Hispanic population. So what you're trying to do with getting all of these genotypes at the various loci is making it so that there's a very, very, very small chance that anyone else in the population would have that exact genotype at all of the different loci. So the suspect, Paul Eugene Robinson, was a 31-year-old ex-con who was arrested in November 1998 for violating his parole. His court records show that he was free after serving part of a 64-month sentence for a string of burglaries. He was caught prowling and loitering around private property. He had previously been convicted of spousal abuse, so blood and saliva samples were taken and sent to the State Department of Justice Crime Lab in Berkeley. So on September 15th of 2000, 3 weeks after the John Doe warrant was filed in Milwaukee and Robinson's DNA profile went out to the lab, the Sacramento police received a call from a state criminalist informing him of a cold hit. So the DNA in Robinson's samples taken at the jail matched DNA that was in the state's rapidly expanding DNA database. So using DNA and the DNA profile, they were able to convict this individual for committing several rapes. So in another case, in 1996, Gerald Parker was charged with the rapes and murders of 5 women between December 1978 and October 1979, as well as the murder of a fetus during a rape in 1980. DNA samples were obtained from the crime scene and were run through the database. 4 cases were committed by Gerald Parker based on the match of the DNA profile. Once the DNA linked Gerald Parker to the 4 different rape cases, he ended up confessing to the crimes. He actually also confessed to a 5th crime in which another individual had been convicted for and had already served 16 years in prison. So the person that had already served 16 years in prison was exonerated and let out of prison, and Gerald Parker was sent to prison for his crimes as well as the crime that another individual had been wrongly convicted of.